Today we are going to talk about ABO blood group system. Remember, there are so many blood group system, right? There are more than 30 major blood group system and there are hundreds of the minor blood group system. But as far as clinical relevance is concerned, two blood group systems are most important. Two blood group systems are most important. Number one is ABO system and number two is RH system. Today we are going to focus and work on ABO blood group system, right? Now, to make a very fundamental concept that what is this group, I will start a simple example that we have a person here who has blood group A, and we will talk about why we call that person with blood group A. Baized, you come here. Now this is a man, and of course I'm sure he's a man, so his blood group is blood group A, right? Now when we say that he has a blood group A, it means if I draw his circulatory system, if I draw his, this is his circulatory system, if I draw his circulatory system should have such type of red blood cells. Red blood cell can be drawn like this or like this, depending upon you're looking from the side or from the top. If I say this person has blood group A, it means that his red blood cells have special antigens which are called A antigens. What are they? A antigens. His red blood cells are loaded with antigens. Yes? A antigens, right? Of course, I will explain very well what is meant by the A antigens, right? And if I say there is one more person and his blood group is B, right? If I say there is another person, let's suppose we talk about Mr. Azaz. Yes, come here. He claims that his blood group is B. And if his blood group is B, now you can understand easily, then his red blood cells should be having what kind of antigens? B, right? He should have which antigens? B antigens, right? So it means those people whose red blood cells have A antigens, they are considered blood group A, and those persons who have B antigens on their red blood cells, they are considered blood group B. I will explain in detail what is A antigen and what is B antigen. And then we take the third person, and this person has A and B, both antigens present on his red blood cells. That this person has A antigens, and not only A antigens on the RBCs, but this person is having B antigens as well, right? So he has both categories of antigens on all his red blood cells, and this person should be called which group? AB group. What is this called? AB group. So here is A group, this is, yes, B group, and this person is AB blood group is mine blood group. So we can take my example, and this is AB blood group. Then we come to a situation, yes, we have one more man here, and he claims his blood group is O. And if we think that his blood group is O, then which antigens are present on his red blood cells? A or B? Which antigens are present? No antigen, simply no antigen. What do you think?
Neither A nor B.
Yes, this person's red blood cells, they don't have A and they don't have B, but they do have something else. They are not really bald like me. They have something else. What is that? They are not naked red blood cells. Of course, they don't have B antigen, they don't have A antigen, but still they have something. They have anti-A, anti-B. He made a mega mistake. Remember, blood groups are decided according to the antigens, not antibodies. So don't tell me anti-A, anti-B. That will talk later. That will talk later. But blood groups are basically named in ABO system. In ABO system, blood groups are given the name according to the presence or absence of A or B specific antigen. If only A is present, A group. If only B is present, B group. If both of the antigens are present, these are antigens. These antigens are then AB group. But what antigen is present over here? Yes, have a seat. And anyone who can tell me here? Yes, so many intelligent people sitting here. I agree with you that this person who has blood group O, he does not have A antigen, he does not have B antigen, but he still has something on his RBCs. His RBCs are not naked, not bald, or we can say not nude. They do have something on them. So what is that? O antigen. I think I will refer to it as H substance. H substance or H antigen. Now I need to explain. Now H antigen has a very, very intimate relationship with A antigen and B antigen. Let me explain. First of all, I will explain what is H antigen. Then I will explain how H antigen in the red blood cells is converted into A antigen in some people and converted into B antigen in some other people, right? So let's come from where these antigens are originating. Let me take a big red blood cell. Okay, I will come over here. First I will explain the H. This is precursor of red blood cell. This is a precursor of red blood cells, because to make such antigens we do need enzymes, and enzymes and proteins are derived from the activation of genes, DNA and RNA, and then protein synthesis, right? But you know that RBC do not have nuclear material. A mature RBC does not have any nucleus. These are anucleated cells. Is that right?
That's right.
If RBCs are anucleated cells, how they can produce such antigens? Answer is that these antigens, either H antigen or A antigen or B antigen, they are made on the surface of precursor cells, erythropoietic precursor cells. When erythropoiesis is going on, those cells, those like erythroblast, and then there is series go into normoblast and different series, eventually reticulocyte and then red blood cells. These precursor cells, the precursor cells of red blood cells do have nucleus. They do have nucleus. Let's suppose this is a precursor cell. This is a precursor cell, suppose erythroblast or normoblast, which is eventually going to convert this red blood cell with H substance. So I will tell from where this H substance comes. Right now I'm going to explain from where this H substance comes. Let's suppose here is your nucleus and there are different type of chromosomes there, right? Now, first of all, there is synthesis of oligosaccharides which are planted on the surface of cells. What are these? Oligosaccharide. What is the meaning of oligosaccharide? Oligosaccharides are short chains of monosaccharides, of simple sugars. For example, this is an oligosaccharide. There's one sugar, then another sugar, then another sugar, and then another. What is this? Monosaccharide. When somewhere between 2 to 10 monosaccharides, when they are made a chain, this chain is called oligosaccharides. Now, first of all, RBCs, all RBCs, right? They are having these oligosaccharide precursor molecule. What is this? Precursor molecule. And there are millions of such molecules on each red blood cells. About 2 millions. The millions of such molecules on each red blood cells. But for explanatory purpose, I will just mention 1 or 2 molecules. So I'm showing it here that here are 2. What are these? Oligosaccharides, right? And these oligosaccharides, which are expressed on the surface of the precursor cells of the red blood cell, they are modified. They are modified by the help of a special type of chromosome, and this is chromosome number 19, and from there a gene expresses. a gene expresses on the chromosome number 19, and this special gene which I'm referring to is going to make a special type of enzyme, and that enzyme is, yes, this enzyme is called Foucault's, pronounce it rightly, Foucault's transferase, right? Be careful. Foucault's transferase. What is Foucault's transferase? Foucault's is a simple sugar. So this enzyme can take a simple sugar, which sugar? Foucault's, and transfer to this oligosaccharide. So what happens if this enzyme expresses? If this enzyme's expressive, I will call it Foucault's transferase, right? This enzyme expresses, it will attach the which molecule? Foucault's molecules with this chain, and this is Foucault's. What is this? Foucault's. Now this oligosaccharide chain with Foucault's is called H substance. What is it called? H substance or H antigen. Is that clear? Now this expression, this mechanism happens in everyone's red blood cells. All of you, either you, it doesn't matter, you are blood group A or B or AB or O, whatever your red blood group, you are going to make first H substance. You are going to make your erythropoietic precursor cells are going to make what? H substance, right? So it means that I can say that here there are H substance. Now you understand what are H substance. We can call it H antigen also. Is that right?
That's right.
Now I can make it like that. This is H and H, right?
Yes.
Okay. This happens in all of you, right? During erythropoiesis. So it means all of us red blood cells on the membrane do express H substance. Is that right? Now, stop the story here. Come back to blood group A, right? Our friend who had blood group A. In his blood, in his synthetic machinery, during erythropoiesis, goes one step further. What is that step? I said, first of all, which chromosome was there? Yes?
19.
19, and that chromosome number 19 was making? Foucault's transferase. It was making Foucault's transferase. So in this person, in this red blood cell, there is oligosaccharide with Foucault's. Clear? But there is one additional activity here. What is that additional activity? We need to go to the chromosome number 9. Chromosome number 9. How many chromosome number 9 you have in your one erythroblast, suppose? Normally in a cell, in a given human cell, standard typical human cells, how many chromosome number 9 you have? Yes, ma'am. 2. One is coming from father, another is coming from mother. Both are not coming from father. So what happens, as you know, most chromosomes from 1 to 22 are autosomes, and X and Y are the sex chromosomes. Now chromosome number 9 is autosome. Autosome 1 comes from mother, another comes from father. So come here. So this young man, I said he has blood group A. Now what he is doing, please do, what is happening in his erythrocyte precursor cell, chromosome number 9, one which came from mother, which came from mother, and other one came from, this is maternal chromosome, and other came from father, right? Now, these two chromosomes at a special locus here, at a special locus at the long arm of chromosome number 9, right? They have a gene. They have a gene, and this gene, when it expresses, it makes a special type of transferase, and that transferase is, yes, who will tell me? Who will tell me? Anyone? It is not Foucault's transferase. There's another type of transferase made here that is called, yes, anyone can tell me what is the name of that transferase? Okay, this is that transferase. Yes. This transferase is an enzyme which can transfer N-Acetyl Galactosamine, Now, at chromosome number 9, there's a genetic locus. At this specific locus, right, when this gene is expressing in person with blood group A, this is going to make which enzyme? Yes, N-Acetyl Galactosamine transferase activity, right? So, at the top of this person has already made H substance. At the top of that, it will transfer, we can call it NAGA, N-Acetyl Galactosamine transferase. So what happens? Here we have added a special top on this. This special top, this whole molecule now is called A antigen. It is called A antigen. Now you understand what is A antigen? Yes. A antigen is produced by addition of N-Acetyl glucosamine to the H substance. Or we can say when H substance is added N-Acetyl glucosamine by the help of this transferase, then H substance is converted into which antigen? A antigen. Right? So it means all those people who have blood group A, like him, all those people who have blood group A, they initially produce H substance.
That's right.
And then do transfer activity of a special terminal sugar, and that is N-Acetyl galactosamine. And then this whole molecule is called A antigens. So we can say that all of these, where I'm putting A molecule, I can enlarge here, right? And if you look at it, what is it? This is oligosaccharide, and here was what it? Fucose. Both of them are making H substance, and at the top of that, what we have added? NAGA, or simply call it N-Acetyl galactosamine. This activity is done by a transferase, and this transferase, because this transferase is converting H substance, this transferase is converting H substance into antigen A. So we call it we can call it transferase A. A simple name can be, okay, I will write it here. Simple name can be transferase A. Is it clear from where the blood group A comes? Yes. You can go back. Now let's talk about the blood group B. Yes. So this friend has blood group B. And if he has blood group B, we come to this story. It means he has B antigens. Now, how is his red blood cells have B antigens on their surface, but from where the B antigens come? Story is somewhat similar. We take a precursor red blood cell, right? As you know, you activate chromosome number 9, and that will lead to the formation of fucose, transferase, and oligosaccharides, right? They will have on their surface, on the side, attachment of fucose, and of course many of these molecules will be made oligosaccharide with fucose, and this is H substance. After making this, then his which chromosomes will be activating? Maternal or paternal?
Both.
Yes, both can be activated, but just a minute. This is again the locus. Now remember, this special point, let me make it, it's a star point. This star point on this chromosome, you know, one chromosome has many genes.
That's right.
And specific genes are present at special location, and this special location is called genetic locus. This is called genetic locus. Now, in this person with blood group A, at this genetic locus, the gene which was present was responsible to make transferase activity for N-Acetyl Galactosamine. But at the same locus, at the same position, in this person, there is a variant of gene, not exactly this gene, there is a variant of slightly varied genes, which makes a different type of transferase, let's suppose this makes a transferase which transfers. This transferase is transferring not N-Acetyl Galactosamine. It's not transferring N-Acetyl Galactosamine. It is transferring only galactose. It is transferring here only galactose. What is this? Galactose. So, whatever H substance are produced here in this person due to the production of
Variant.
no, due to the production of which transferase? Galactose transferase. We are having galactose attached over here.
That's right.
Right? And once to the H substance, galactose is added. When on the H substance, galactose is added, this is called B antigen. What is called? This is B antigen. That is why this transferase is also called B transferase.
That's right.
Right? Now let's compare both of these persons. Come here. Now what we can say, you can come here. Now we can compare. This is blood group A, this is blood group B. Both of them do make H substance. Both of them on the red blood cells do make H substance. And once they have made H substance, this person produces a transferase A which transfer N-Acetyl Galactosamine to his red blood cells H substance, and thus it generates antigen A. And that is why his blood group is called blood group A. But in this person, what is happening? That in this person, first again H substance is produced, but because of there is presence of a different transferase here, which transfers galactose on the galactose or galactase, we can see it. Galactose on the top of, what is this? H substance. So this will convert into B antigen. So now you clearly understand the difference between the person with A blood group or with A antigen and person with B the blood group with B antigen. Is that clear? Please have a seat. Here, I want to make one more story clear before I move further. Person who has blood group A, he has A antigens here. Now, this gene which produces transferase A, this gene is present on maternal chromosomes or paternal chromosomes. It may be present on any one of them or may be present on both. Before you tell me something totally new, let me tell you. This gene which produces transferase A. It may be present in only one chromosome. Either it is coming from mother or from father, or it may be coming from both sides. So it means that it is possible that all those people who are having blood group A, some of them are having this gene on both chromosomes. In this case, they are producing high amount of this enzyme, and they are rapidly converting the H substance into A antigen. Is that right? Now phenotype is the expression of gene on the producing a trait. Phenotype of this cell is A, but genotype is AA.
Yes.
One A from maternal side, other from paternal side. So one person may have blood group A because his maternal and paternal both chromosomes contain a gene which is producing transferase A. But this is also possible. But just remember this. Later on I will go into further detail. In this, I will go into further detail. If one person has on one chromosome functional gene and other gene is not functional, other side, the same locus gene is not functional, right? I will tell you what is this non-functional gene later. Again, one person, both sides functional, he is producing a lot of transferase A, done. But you may come across one more person in which one chromosome has active gene which is producing transferase A, other is non-functional gene. What will be his blood group? Still blood group A, because even though he will be producing less transferase, but still that less transferase will be enough to convert all the H to A. It means genotype can be AA, maternal A and paternal A, or there may be AO. This A from maybe maternal or paternal, O may be from other partner, other parent. But you need to remember, when there are two A, blood group is A. But even if there is one A, still blood group is A. It means all those people who have blood group A, all those people in this world who have blood group A, if you study their genotype, genetic material here at this locus, some will have both sides same gene. We call them they are homozygous for A. And some will have one side at one locus of chromosome what? Active A, other side there may be inactive non-functional gene. Then we call it AO. And this is not homozygous, this is heterozygous. So people who have blood group A, if you look at their genotype, it may be homozygous A or heterozygous A. Is that clear? Now we come to this thing. We came here. Now, here was transferase A gene and here is transferase B gene. This was a person who is going to make blood group B. What is this gene? Transferase B. Now, let's suppose he has transferase B here on maternal side, as well as he has transferase B gene on which side? Paternal side. If both of them are producing transferase B, heavy amount of transferase B is produced, which is rapidly converting H substance by addition of galactose into antigen B. But it is also possible that from one parent you get, if both parents you get, what is this? BB. Or it is possible that from one parent you get active gene B, or other side at the same locus there is inactive gene. So this will be BO. You are understanding? So in this case, all the people in this world who are having blood group B, some of them will be BB when genotypically they will be BB, because from mother and father, both sides, they are receiving active genes, but some will be BO, where from one parent they are receiving the active gene, that is for transferase B, and other side is having inactive gene or non-functional gene. Am I clear? So how many genotypes? Genotypes are there for blood group A. There are two genotypes. Phenotype is A. Genotypes are AA or AO. People who have blood group B, how many genotypes they can have? BB or they can have BO. This person will be homozygous and this person will be heterozygous. Is that clear? Now we come to person like me. My blood group is AB. It means there's double action going on, right? What is that double action? First of all, the basic activity. Chromosome number 19 making Fuco's transferase, and these oligosaccharides, and this oligosaccharides, they are having what? Fucose. H substance. Clear? After that, I have one chromosome from my mother and other chromosome is from my father. Right? Maternal chromosome is, and of course there is a little lipstick too. And here is father chromosome. You can understand what is it? A little, right? If on the same locus at chromosome number 9, let's suppose this is that locus, same locus, here there is the gene which is going to make transferase A, that is, and from here, from maternal, okay, it doesn't matter from which side A is coming and from which side B is. Suppose paternal chromosome, that is making transferase B. Transferase B. So in my precursor red blood cells, one parent chromosome is making transferase A and other chromosome, parent's chromosome is making transferase B. So both of them are active. So what will happen to these H antigens? In some people, it will be converted into antigen A, and in other persons, and other molecules, some H molecules will be converted into antigen A by this action, and other H molecule will be converted into antigen B by these actions. So my red blood cells will have both kind of molecules, A antigens as well as B antigens, and my blood group will be AB. Right? And how many genotypes I can have? Only one. Don't tell me 0 genotype. Who said this then? I have one genotype because it is possible from father A and mother B side, or it is possible father is giving B locus, B, and mother is giving A, but in both cases it's AB. So here is BB and B0, BO, here is AN, AO, here is AB, and then AB. So there is only one type. You understand it?
Yes.
So how many underlying genotypes we have learned? Genotypes. For blood group A, we have 2 genotypes. For blood group B, we have 2 possible combination. For blood group AB, we have only one genotype. Now I want to ask one more thing. Okay, let's go to next person. This is blood group O. What happens in this person? That he is producing H substance, as I told in the beginning of the lecture. But the issue is this, that at chromosome number 9, at the same locus, okay, I will show it by green color, the non-functional gene variant. Non-functional gene variant. It has on both sides non-functional gene variants. It means, actually it is mutated. To be very true, now, these two are actually, during the evolution of humans, somewhere it got mutated, and due to mutation, they don't produce active products. So they are non-functional or minimally functional. So even though these genetic genes are present over there, but they cannot produce transferase A, and they cannot produce transferase B, and whatever they are producing is not functional. So in such patient, can H convert into A antigen?
No.
Can it convert into B antigen? No. So it means there is zero change in them. There is zero change in H. Because it is zero, so some people started writing this blood group is zero. And then other people modified the concept. They read zero as O. So they say this blood group is O. So what is blood group O? If I say, come please here. If I say this person is having blood group O, what's going on in his erythropoietic process as far as ABO system is concerned? What happened there in his system, that chromosome 19 was able to attach the fucose to the oligosaccharide and produce H substance. But from maternal and paternal, both sides, he received the genes which we call it, yes, what we call these genes which are inactive genes. They do not produce any functional transferases. So H substance could not be converted into A antigen or B antigen. So they stay the H antigen or H substance, right? Is that clear? And such people who are having only H substance on their red blood cells, of course they do not have A or B antigen on their red blood cells. Such person's group is called O group. Please have a seat. Any question up to this? Now, let me make it some terminology more clear. This is chromosome number 9 in all examples. But we have seen, and at this very special star point, or you can say it's special locus, sometimes a gene is there which can make transferase A, right? Sometimes there is gene which is transferase B, and sometimes there is gene which is non-functional. It means at one locus there can be only one gene. You just imagine, now listen carefully. Just imagine, here is this locus is like a cup, ice cream cup. Either you put what here, chocolate flavor, or you put vanilla flavor, or you put ice cream which is tasteless, no taste.
Sugar.
Is that right? Tasteless ice cream. So this position, gene may be having for transferase A or transferase B or non-functional. Is that right? Now what is the relationship of these genes with each other? What is the relationship of these genes? These genes are called alleles of each other. These genes are called alleles of each other. You have heard the word allele too many times. What are alleles? Alleles are variants of one gene. Variants of one gene, like variant of ice cream. Now there can be many flavors of ice cream. In the same way, one gene may have many variants. As long as that gene is present on the same locus, same position of the chromosome, right? For example, I have one chromosome. Okay, let me tell you more clearly. Oh, I don't have hair, but I will give example of hair. What do you think? I should give example of blonde hair and black hair example, or do you want me to blue eyes and brown eyes example? Tell me, I will explain the alleles. You want about eyes? Okay. Suppose there are two chromosomes. They determine the color of the eye. One chromosome is coming from mother, other chromosome is coming from father. Is that right? At a very special location, suppose at this point and this point, these locus are same on fingers. So we call them homologous positions. And these, if this is chromosome number, whatever number is this, it is the same number of chromosome. For example, if this is one chromosome, this is also one. Then they are homologous chromosome. If it is chromosome number 10, this is also chromosome number 10, they are homologous chromosome. I'm just trying to teach you a little basic, right? On the homologous chromosome, at the same positions, at the same locus, whatever genes are present, if they are variant, we call them alleles. Is that right? Now let's suppose, if there is, go finger or diagram, where you understand better? Diagram, okay. Let's suppose these are two chromosomes. And this is a special locus here and here. This locus is going to tell the color of the eye. It determines what kind of pigment can be produced and what should be the concentration. Now, at these both locus, one is coming from mother, other is coming from father. For example, from mother side, this is maternal and this is paternal. From mother side, it becomes brown color. And from father side, also it becomes brown color. So we say mother and father both have given alleles. If this person has inherited the alleles from mother and father both sides, for which color? Brown color. His eyes will turn, baby eye will turn brown. Now another baby, here one parent is giving for the brown color, other parent is giving the allele for blue color. What will be the color of the baby eye?
Brown.
Or one will be brown and other will be blue, or half will be brown and half will be blue. Yes, what will be the color of eyes?
Brown.
Depends upon dominance.
No. Okay, then you must know which gene is dominant, blue or brown?
Brown is dominant.
Brown is dominant. So what happens? If both are brown, baby eyes are brown. But if one is blue, other is brown, brown is the dominating. Brown is the dominating gene. So still baby eyes will be brown. Here genotype will be blue and brown, but phenotype will be, again I'm sad, brown. Is that right? Here genotype is brown and phenotype is also brown. But now we imagine another baby which inherits maternal allele for blue and paternal allele also blue. Now what will be the color of eyes? Blue. What did we learn here? We learned this thing that if one allele is brown and other allele is blue, then brown will dominate and show its effect, and blue is recessive. Blue is recessive because in the presence of brown it becomes suppressed and it does not make blue eye. Is that clear? Blue colored eye, I must say. Is that right?
That's
Now we come back to this. So if you really want blue eyes, then both of them should be, mother and father both should bring which color? Blue. Is that right? Now we learned this is dominant gene and this is recessive gene. Now we come over here. Here I said, if there is A allele, what is this? A allele, other may be A or may not be A. Other may be A or may be non-functional What will be the group of blood? A. So it means A is dominant or recessive? It is dominant. In the presence of O, okay, we call it O. In the presence of O, A dominates. So we call it autosomal dominant gene. And this O will be recessive. This is recessive. Now we come again. So that is why people who have blood group A, they have both A or they may have one A. But because A gene is dominant, then O will not have influence and still blood group will be A. Is that right? Now we come to next example. Even B is also dominant. Why? Because if B is present with B, of course, B group is there. But if B is present with O, still blood group will be B. So it means B is also dominant in the presence of O, which is non-functional. Clear? But now come over here. One parent give A allele for transferase A, other parents give allele for transferase B, right? Now both of them are able to express. So none of them is recessive and both of them are dominant. Both of them are expressing. So what we can say here, this is codominance. Codominance. You know, when husband and wife, both of them are codominants, they are fighting all the time. Because both of them want to have their expressions. If one partner is recessive, other is dominant, then system is played very easily. Right? But whole structure of the family will be according to the dominant partner. And recessive will be just agreeing to everything, whatever is happening. But if both of them are co-dominant, trouble will come. But let's come to what is happening here. Both of them are actually, O is recessive in the presence of B. O is also recessive in the presence of A. But if so none of them could be O. But when both of them are together, like blue eye situation, if both of them are together, then O trait is shown. Blood group will become O. So all the babies in this world who are born with blood group O, their genotype must be O. Both sides? O. Is that right? All babies which are born with blood group AB, their genotype should be AB. But all the babies who are born with blood group B, their underlying may be BB or BO. And all babies who are born with A, right? They will have underlying genotype either AA from both parents, they are bringing the allele which is A, or AO, right? Any question up to this?
Clear.
Clear. Okay, let's have a break. Thank you, Ji.